In BT and its applications for medicines, today we will be learning about gene therapy. Already you have learnt about molecular diagnosis, that is, how do you detect a sort of a disease by using PCR. Then we had also learnt about ELISA, enzyme linked immunosorbent assay technique, and the third important applications primarily here relates to gene therapy. Now, what exactly is the meaning of the term gene therapy? When you take gene therapy is a corrective therapy. It is a corrective therapy with relation to treatment of human hereditary diseases. Human hereditary disorders can also be due to chromosomal disorders. So, we are using genetic principles of treatment, and we are also using the tools and techniques of molecular biology. That is, we are using recombinant DNA technology, that is, we are using genetic engineering by restriction enzymes, gene cloning, etc. We are using tools and techniques of molecular biology and adding to that the genetic principles for correcting the sort of a chromosomal disorders or human hereditary diseases which are inherited. So, here what do you do in a sort of a gene therapy? You insert a gene. So, when you insert a gene, you are primarily countering the gene, the defective gene, or you are correcting the sort of a defecting gene. We already know there is a presence of a defective gene. So, once there is a defecting gene, we are countering it, we are trying to change it, correct it or counter it, and we are trying to insert your sort of a gene of interest. When you insert your gene of interest will show newer metabolic function. It will show newer metabolic function. So, where exactly do you practice gene therapy? You introduce your normal gene in normal cells and tissues of the human being, or else you introduce it into the embryo. You got three situations: cells, tissues and embryos of humans. And what is the basic principle here? The basic principle is you follow the antisense technology. Already you are familiar with the antisense technology with relation to the sort of nematodes. You follow the antisense technology. Thereby, we can exactly, if supposing a patient of HIV or AIDS patient has got a low immunology, we can correct it. And primarily gene therapy is by introducing a gene, you change and you enable a particular human being to have a new metabolic function. So, sickle gene therapy is exactly, it has got applications in the treatment of cystic fibrosis, muscular dystrophy, sickle cell anemia, and also hemophilia, wherein primarily these are all very famous diseases which have encountered with famous popular human diseases. So, you can have a sort of a solution to all these particular things. Now, the most important application of gene therapy relates to adenosine deaminase deficiency or ADA deficiency, which is seen in small children. So, in 1990 we had a breakthrough because we had the first gene therapy in humans with relation to ADA deficiency. Now, ADA deficiency is a severe immunological deficiency. Thereby, when you study gene therapy or when you study molecular diagnosis, as well as when you have certain basic biochemical techniques like enzyme linked immunosorbent assay technique like or ELISA, wherein an antibody and an antigen binds, you can very easily take the sort of an antigen and also bind it to a particular sort of a molecule and target it to a particular sort of a cell. So, thereby we understand that along with newer tools and techniques in biotechnology, even BT has got lot of applications with relation to medicine in terms of gene therapy and also in terms of curative genetics. So, applied genetics or applied human genetics and medicine are both blended together with relation to the basic principles of biotechnology and molecular biology. Thus, we understand an integrated knowledge of cell and molecular biology, biochemistry, cell physiology, the cell pathology and the sort of physiology, all of them integrated together form different ways of how you look at solutions to biotechnology.